part of the core of your body, and that's because it's constantly being cooled by outside air. And if the outside air is cooler than 37 degrees centigrade, it's going to cool your lungs off. All right. Now, just follow along with this. The right side of this equation is relatively constant. If you are, say, close to sea level on Earth, the number of gas molecules in the atmosphere is relatively constant. The gas constant is constant, and your body temperature is constant. So if the right side of that formula is relatively constant, that means pressure and volume are inversely proportional. For pressure times volume to be equal, if one goes up, the other has to go down, and that's inverse, being inversely proportional. Okay? Now, how does this relate to our physiology? This relates to the mechanics of breathing. They're in red. So you can kind of remember that those three names are the same: ideal gas law, universal gas law, or Boyle's law. But it relates to the mechanic of breathing. Basically, your muscles of inspiration, they contract to increase the volume first. When you're breathing in, your chest expands, the volume increases. And because of the ideal gas law, if volume goes up, pressure has to go down. They're inversely proportional. And the pressure drops below that which is in the room. And air moves just like everything else we talked about with passive movement. It's going to go from high to low, except it's going from high pressure to low pressure. This is what causes wind to be outside. You have certain areas with higher air pressure and certain areas of lower, and that air is moving from high to low. And that's basically what's happening here. So you drop the pressure inside your lungs below the room, and it goes from the room into your lungs. Okay? Then when you decrease the volume, that increases the pressure above that which is in the room, and that's how you exhale. This is about the mechanics of ventilation. Slide 33 is just showing you the figure from your textbook, and it's basically showing you, and I've just kind of labeled it to the left, how the volume changes first, that drops the pressure, that moves the air in. Okay? So again, all these three names are the same. So the muscles that are expanding the volume are the muscles of inspiration. And this should have been covered in anatomy. But you have to think about ventilation. Remember, ventilation is the moving of air. A lot of patients and lay people will call it respiration. Just let them. But you got to know that's not respiration. We talked about respiration when we talked about aerobic cellular respiration. But this is ventilation. And when you're studying ventilation, study it in two ways: quiet breathing and forced breathing. That's the two columns on slide 34. So on quiet breathing, you've got three sets of muscles contracting on inspiration. Diaphragm drops the floor, external intercostals widen the chest cavity, and then up here in the anterior is the interchondral portion of the internal intercostals. And you might think, well, internal intercostals should be drawing it in, not these particular intercostal muscles. Where the cartilage, where the ribs are cartilage right next to the sternum, it actually lifts the sternum out. So you got external intercostals doing this. You got the interchondral portion of the internal intercostal muscles doing this. And then you got the diaphragm dropping the floor, and that is what is increasing the volume. For quiet breathing, eupnea, you're not really working on breathing out. You're just shutting those three muscles off. And the passive recoil of the lung, and it has to do with all the alveoli having surface tension, that actually causes the volume to go back down. Now, if you're running around the track, you are going to use those three muscles on inspiration that you used in quiet breathing, but then you're going to recruit neck muscles. You're also going to recruit some other muscles to some degree of variation, pectoralis minor and serratus anterior. But if the diaphragm drops the floor, the neck muscles raise the roof, and you get a much bigger increase in inspiration volume. And then breathing out, now that becomes active. You got the interosseous portion of the internal intercostals. That first bullet on the bottom right box, you should draw an arrow between that and the external intercostals, because those are muscle antagonists. External intercostals do this. The interosseous portion of the internal intercostals does this. Those are muscle antagonists. Again, the diaphragm drops when it contracts. To lift it up, to blow air out, you have to shut it off and then contract underneath it, and that's all those abdominal muscles. And if you remember from anatomy, along the sides, they're extremely thin, the thin three sheets: external oblique, internal oblique, and transverse abdominis. Extremely thin. And that's why if you're out of shape and you're asking them to contract every breath out, they're going to cramp up and you get a side ache. Slide 35 is just the same thing as slide 34, except it's in picture form. Okay? Kind of tells you what some of the actions are too, and the direction of the pull. The next slide, 36, shows you how the interchondral portion of the internal intercostals actually causes the sternum to lift out forward. And that's on the bottom left of that figure. Okay, let's move on to the next law, and that's Dalton's law of gases. This is a little bit different. To understand Dalton's law of gases, you have to understand that we are like those ugly-ass fishes that live on the bottom of the ocean. Those ugly-ass fishes are born in extreme pressure. They live their whole lives in extreme pressure, and they die in extreme pressure, and the whole time they didn't even feel the extreme pressure. When we live here on land, near sea level, the same thing. We are born in this tremendous air pressure. We live our whole lives with this tremendous air pressure. We die in it. We don't even realize it's pushing on us. This is the best way to imagine it. Think about just your skin. Now imagine a square inch of your skin. There's actually 15 pounds pushing in on that single square inch on your skin. Now, you're covered with skin. Each square inch is being pushed in by 15 pounds of pressure. And that's just gravity working on the atmosphere. And that's why I have these pictures on the slide 37. Gravity is pushing on the entire atmosphere right up to outer space. And as it's pushing down all those tens of thousands of feet of air, by the time it's on sea level, it is a tremendous pressure. You just don't feel it because you're like those ugly-ass deep-sea fishes. So if you ever see a movie, a science fiction movie, where someone is out in space and all of a sudden their spacesuit gets a leak, first of all, they're probably going to freeze and become solid pretty quickly. But if they don't, they explode. You remove the 15 pounds of pressure on every square inch, poof, you just pop. So there's a few movies that actually show that, which is really gross. But anyway, so you see this whole column of air pushing all the way down to sea level. And if we have a vacuum tube with mercury, which is liquid metal, this metal, the melting point is such that at normal temperatures it's liquid, but it's heavy because it's metal. It will actually lift the column of this heavy liquid metal 3/4 of a meter. You remember the meter sticks? Well, 3/4 of it, this is how much pressure there is at sea level. It'll actually lift that up. And just a little over 3/4. It's not 750, it's 760 millimeters of mercury. That is the total pressure of the entire atmosphere at sea level. Now, Dalton's law of gases is basically about how we describe a mixture of gases, because there are multiple gases in the atmosphere. And in Lab 11, if we were talking specifically about the chemoreceptors involved in volunteers running around the track. By the way, do the homework for Lab 11, because there's going to be questions on the quiz next week regarding that homework. But this is why when we talk about the partial pressure of things, and we're talking about Dalton's law of gases. And so if you ever see the insignia PO2 or PCO2, the little p stands for partial pressure. Okay, so if we look at the atmosphere as being a mixture of gases, the total pressure is 760 millimeters of mercury. And then you put a little p in front of all the different gases, and that's how all the different gases mix together to form this whole atmosphere. Now, the vast majority of our atmosphere is N2 nitrogen gas, which is inert. It does nothing. And that's probably one of the reasons why we have life on this planet, is because most of the atmosphere is inert. Okay? And you can see here, slide 37, what the numbers are. The partial pressure of O2 at sea level is 159 millimeters. There's very little CO2, and that's why we can measure the difference, because it doesn't take much additional CO2 to see that tiny number rise. And then look how the vast majority of the atmosphere is nitrogen gas. Can anybody guess why that's really important, that most of our atmosphere is inert? Nothing. Okay, look at that number for oxygen. 159 millimeters of mercury. If it's 160, fire season is pretty much all year round. Okay, that's why it's really important that most of this atmosphere is inert, because if you get more oxygen than that, fires, you just would not be able to put fires out. Okay, now there's other implications for physiology besides that, with Dalton's law of gases. And some of it has to do with what happens at different pressures. If we're at sea level and the partial pressure of O2 is 159 millimeters, if we go up into the mountains, the whole total pressure goes down. That's why your ears pop when you go up into the mountains. But that means all these numbers go down proportionally. So if that 159 for partial pressure of oxygen, if that goes down, your kidney cells are going to get less oxygen. They're going to release more EPO. Hopefully this is ringing a bell. And then you're going to get more erythropoiesis, and pretty soon your hematocrit, your percentage of blood occupied by red blood cells, goes up. And that's physiologic polycythemia, as it says there in the first bullet, slide 38. Okay? But we can also go in the opposite direction. And this is what happens when people do scuba diving, or more specifically, deep sea diving. As you go into the water, deep sea diving, the pressure is all about you. And the pressure is also in the gases that you're breathing. If you ever wonder how they were able to get bubbles in champagne or Diet Coke, they push high pressure of CO2 in water, and it pushes the gas into solution. At that high pressure, it doesn't look any different until you release the pressure, and then it starts coming out of solution and becomes gas. And that's why Diet Coke and beer and champagne have bubbles. It was pushed in under high pressure, now it's coming out. Well, if you go deep sea diving, and this is the second bullet on slide 38, it pushes the gases into the plasma. And that's why when you come out from deep sea depths, you've got to come up slowly. It's like these soda machines here at the college, you know, it's like somebody who designed it is a practical jokester because you put your money in, right? And it goes boom, 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 boom. Well, then you get the Diet Pepsi out. You got to sit it on the table for a while. You can't open it for a few minutes. It just went boom, 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 boom. Because if you do, it's just going to squirt all over. That is the bends. If you come up too quickly from deep seas, it's like boom, 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 and all these bubbles start appearing in your plasma. And you're not supposed to have bubbles in your pipes. It's supposed to be all liquid. And that kind of stuff causes problems in capillaries. So deep sea divers have to come up gradually, and then you can, it's just like with the Diet Pepsi, you have to, and then you have a mess. Okay? So that's decompression sickness or the bends. You got bubbles. All right, now let's talk about oxygen transport in blood. For lecture exam 4, you're going to need to know the 2 forms of oxygen in arterial blood and the percentages, and they're right here on slide 39, so pay attention to this. The vast majority is actually hooked up to heme groups on the iron in hemoglobin, 98.5%. This is what I've been telling you the whole semester. There's nothing black and white, even how much oxygen is on hemoglobin. It's not 100%. It's still a shade of gray. It's 98.5%, and 1.5% is just dissolved in plasma. Now, when oxygen binds to the heme group, and that's what this animation is on the bottom right here on page 30, slide 39. It actually twists the quaternary protein, and it turns it bright red when it twists. That's oxyhemoglobin. So that's 98.5% of the oxygen is oxyhemoglobin. We got a little review of hemoglobin from lecture exam 1. This is one of the few examples in our body of quaternary proteins. Okay, so we have 4 actual protein chains that have tertiary protein structure. There are 4 globs, which is where the name hemoglobin comes from. So we got 4 globs. Each has a heme group in the center. In the center of the heme group, and in the bottom left, those are the green flying saucers. In the center of the heme group is an iron atom, and that is what the oxygen actually binds to. What you need to do, and also get your lecture packets out and turn to page 65. We're going to be covering these pages in your lecture packet. And look at the graph on the top right. The graph on the top right on page 65. And that is what the second bullet under number 2 is saying. Of the 4 globs, each with a heme group, so quaternary hemoglobin has 4 heme groups. They do not have the same affinity for oxygen. Okay? Each has a different affinity for oxygen. So if you're looking at that graph on the top right of page 65, this one right here, heme group number 1 has the highest affinity. The reason why heme group number 1 has the highest affinity is that with a really low partial pressure, look at this is atmospheric pressure, you're looking at 20 millimeters of mercury, boom, it loads because it has the highest affinity for the oxygen. Heme group number 2 has the second highest affinity for oxygen. At about 40, it loads with oxygen. But heme groups 3 and 4, even with that oxygen, it's not loading. And heme group number 4 on the very top right has the lowest affinity for oxygen because you've got to get all the way up to 100 millimeters of mercury for that last and fourth heme group to load. So hopefully everybody understands that. Good thing these are recorded, huh? Then you can keep replaying it over and over again. Okay, but the bottom line is, if you look at that graph on the top right, the higher the partial pressure of oxygen, the more saturated the heme groups are going to be. Okay? And this is from your textbook on slide 40, and it shows, again, not all the heme groups are loaded, but 98.5 is the normal saturation of the heme groups in arterial blood, 1.5%. It's floating in plasma. And then there's a picture from your textbook showing you the quaternary protein in the heme groups. Okay. Now, we got to get used to a couple of terms here. When oxygen binds to the iron, that is called oxygen loading or association. When it falls off the iron, that's called unloading or dissociation. And so if you just think about, big picture, you're breathing, getting oxygen. In your pulmonary capillaries, the oxygen is loading. Going in arterial blood, 98.5% is now on heme groups. When it gets to systemic capillaries, you don't want those oxygens to stay on the heme groups. You want it to fall off so it can go through to your trillions of cells that need the oxygen. So this is the best way to understand respiratory physiology is in terms of pulmonary capillaries and systemic capillaries. Okay? You want to get oxygen loading and association in the pulmonary capillaries, and you want to get oxygen dissociation or unloading in the systemic capillaries to go to all the trillions of cells. And this is where we talk about the oxyhemoglobin dissociation curve. Okay, and if you look at that first bullet, you will see that nothing is 100%. Nothing is black and white. Everything is a shade of gray. And when you're just sitting at rest, only 50% of the heme groups unload the oxygen. You work out and exercise, and you need more oxygen, and 75% of the heme groups will unload. But that still means that as it gets through the systemic capillaries and goes into systemic veins, there's still some oxygen on some of the heme groups. It didn't unload at all, okay? Now, the topic of the oxyhemoglobin dissociation curve, and that's shown on the rest of the, actually the top left of page 65, is very similar to this graph here. Now, if you look at this graph on the bottom right of slide 41, you will see there are three curves. And this is generally how we look at the oxyhemoglobin dissociation curve, which the center line that's in the middle is the normal. Then you have a line that's to the left of that center line, and that's called the left shift. And then you have a line to the right of that center line, and that's called the right shift. Does everybody see that in that graph? This is the best way and easiest way to think about it. The left shift is what we see in pulmonary capillaries, in your lungs. The right shift is what we see in systemic capillaries, in all your tissues. Okay? And oxyhemoglobin changes depending on the conditions found in either the pulmonary capillaries and systemic capillaries. Hemoglobin is a miracle protein, it really is. But look at those conditions on this graph, on the slide, and we're going to cover this in more detail in the next few slides. So don't think this is all in, but I just want to get you grounded in what the next few slides are going to talk about. So the left shift, you have higher affinity for all the heme groups because of these conditions. Again, they are not all the same. The four will have still different affinities, but all four will increase with the conditions found in the left shift. What are they? You have lower CO2. Why? Because in pulmonary capillaries, you're blowing it out. That means here, there's going to be less. This is connected to, again, the outside world. Every time you breathe in, you're cooling off the lungs. You have lower temperatures in your lungs. You also have, whenever you get rid of CO2, you're going to make things more basic, more alkaline. That has to do with this formula. Remember that this formula, and we're going to talk about this more because this is actually a buffer system, is an equilibrium, which means it wants to be level. If you blow off CO2, you're going to get rid of some acid. That H plus is acid. If you have higher CO2, you'll have more acidity. It goes hand in hand because of this formula. So again, going back to this slide, in pulmonary capillaries, it's cooler. You have less CO2, so it's more basic, alkaline. And then this last bullet is also talking about one of the few processes that red blood cells do, because it makes this 2,3-DPG. And when you get into these conditions, red blood cells make less of it. So you have low levels of 2,3-DPG. You got these other conditions all found in here. And boom, you increase the affinity. And these heme groups are sucking oxygen out of the alveoli and into the red blood cell. Then you go to the right shift. We found in systemic capillaries, you got a lot of cells working. A lot of working cells are making a lot of CO2 and maybe even lactic acid if they're working really hard. All of it is making it more acidic, more work, more metabolism, more sliding filaments. You're going to get higher temperatures. And the red blood cells are going to make a lot more 2,3-DPG. All of that is going to contribute to this right shift. And all the heme groups drop their affinity to oxygen and just let it go. Not all of them, and you see the percentages up on the first bullet, but because of this lower affinity, the oxygen falls off to the tissues that need it. Everybody following this left and right shift? Okay, next slide. On slide 42 is showing you from your textbook the normal. So you might want to write this on this picture from your textbook on slide 42 and just say this is the normal. It's also showing that different heme groups have different affinity. And that's why, because not all the heme groups are going to release oxygen here, you actually do have some oxygen in systemic veins. All right, so when we talk about the effect of pH, that is given a name called the Bohr effect, and that's on slide 43. By the way, this is actually how acid is formed. It's actually not free-floating hydrogen ions. It actually bombs onto water and forms what's called a hydronium ion. Okay? And again, the Bohr effect, if you get more acidity, you're going to cause the oxyhemoglobin dissociation curve to slide to the right. Okay. And that's showed on slide 44 from your textbook. You drop the pH, that means it's getting more acidic, and it's the line, the bluer line to the right. Closely related to this, because of that formula that I had on the center board, it's not exactly the same, but they go hand in hand. If you have more CO2, you have more acidity. But the specific effect of CO2 is called the Haldane effect. And again, if you have more CO2 that you have in systemic capillaries, you're going to cause unloading of O2. Okay? And look at that figure there next to Haldane effect. This is, you can see other writers of textbooks and internet materials, they are concentrating on this distinction. That figure's up on top, pulmonary capillaries, down on the bottom, systemic capillaries. But because the tie between pH and CO2, that's why the Bohr effect and the Haldane effect work simultaneously. They're working hand in hand. And they're causing either a left shift or a right shift. And then the other condition is temperature, and that's described on 46. Again, working cells are warmer, and that's going to be warmer at the systemic capillaries. So that's what I would do here. I'm not going to write it up, but I would just write all those conditions right in a row right here. Temperature, pH, CO2 levels, 2,3 DPG levels. Circle the two that we're talking about, pH, and write Bohr effect. Circle the two that have to do with CO2, write Haldane effect, and you're going to have this down. Okay, so in the box is a nutshell of systemic capillaries. The box on the bottom left is about systemic capillaries at the tissues. Warmer temperatures, more CO2, more acidity. You can also write more 2,3 DPG, and all of that is contributing to the right shift, which is going to stimulate oxygen unloading to the cells that need it. Okay, are we good there? Okay, so there's two forms with percentages of O2 in arterial blood. For lecture exam 4, you need to know the three forms of CO2 transport in venous blood. The vast majority of it is actually right over here. Venous blood. %, is actually not traveling as CO2. It's traveling as bicarbonate ion in plasma. This will go out into the plasma. So in arterial blood, the vast majority of oxygen is in the red blood cell. In venous blood, the majority of CO2 is in the plasma in the form of bicarbonate ion. Okay, and then you see in that box that same figure that I have up here on the whiteboard. Second, 7.2% is actually in the red blood cell. It's not bound to the heme group. It actually will bind to various amino acids of the globin protein. Okay, and we call that carbaminohemoglobin. 7.2%. And then the remaining 5.8% is also in the plasma. But this is not bicarbonate ion. It is actually just dissolve CO2 in plasma, dissolve like Diet Coke. So yeah, the vast majority, over 90% of the CO2 in venous blood is in the plasma. But not just as CO2, only 5.8% is in CO2, 87% is bicarbonate ion. Okay, and you can see those same percentages up from your textbook on slide 48 on the far right column. Okay, and then turn to your, some of this, what we've been discussing about hemoglobin is on page 66 in your lecture packet, including a description of Bohr effect and Haldane effects. Also talks about 2,3 DPG. And then on page 67 is the cardiovascular loop. This is a pretty handy figure that shows a lot of things, and some of it we already talked about in one figure. For example, if you look at the cardiovascular loop, at 12 o'clock, at the very top of that loop is the pulmonary capillaries. That's what I would write up there. Well, it says alveoli, but that's pulmonary capillaries. Then at 6 o'clock, down at the bottom where it says tissues, that's systemic capillaries. The whole right side at 3 o'clock is systemic arteries. And the left side, 9 o'clock, those are systemic veins. So this is the concepts that are talking about here on the cardiovascular loop, where you see oxygen going into the red blood cell, those arrows, that is oxygen loading. Then where you see the arrows up there at the top going out of the red blood cell and out of the blood, that is CO2 unloading. Then down at the bottom at 6 o'clock, you can see the opposite. Oxygen unloading, CO2 loading. You can also see that in red blood cells, it's actually tipping the scales. And the scales are going in this direction. It's not just a straight equilibrium. But red blood cells have an enzyme called carbonic anhydrase, which is kind of tipping the scales to go to the right side of that equation. Also covered in the cardiovascular loop are the 3 forms of CO2 transport and the 2 forms of oxygen transport. And then finally, there's one thing I want to add in here because it is important. Bicarbonate ion is negative, and if it's coming out of the red blood cell into the plasma, unless you do an exchange, you're going to cause there to be a membrane potential, an unnatural membrane potential for red blood cells. And so to prevent that, down here at the bottom at 6 o'clock, you see what's called the chloride shift. In other words, negative bicarbonate ion comes out of the red blood cell and negative chloride goes in. No battery, no voltage, negative for a negative. Then at the top, at the pulmonary capillaries, you see the reverse chloride shift. The chloride comes out and the bicarbonate ion goes back in. This is kind of bizarre. You're just going to have to just be okay with it. But it's to get CO2 to go out into the atmosphere from your body, that bicarbonate ion has to go back into the red blood cell, go in this direction, become CO2. The CO2's got to diffuse out of the red blood cell, out of the plasma, and go all the way into the alveoli. That's just the way it is. And you can actually see that right there in that red blood cell at 12 o'clock in the cardiovascular loop. You can see the bicarbonate ion going back in, the chloride coming out, reverse chloride shift. You could see that formula on the whiteboard going in reverse to CO2, and then you see the CO2 going out of the red blood cell, out of the pulmonary capillary, and out to the alveoli so you can exhale it. Okay, so this is showing you the slide 50, the exchange at systemic capillaries. CO2 is coming from the working cells. It's going into the red blood cell. Carbonic anhydrase in the red blood cell is making it into bicarbonate ion. And then you see the enzyme that's doing the chloride shift. Bicarbonate coming out of the red blood cell, chloride going in, no weird voltages. Slide 51 is showing you the exchange of CO2 at the pulmonary capillaries. You got the reverse chloride shift down at the very bottom with the bicarbonate going in, chloride going out. Then you see going in reverse direction in this equation, becoming CO2, and then the CO2 going all the way out to the alveoli. Okay? Any questions? Cardiovascular loop, we're actually doing pretty good on time. Is it because we're being recorded? You guys don't want your question to be recorded? Everybody's got it? Are you guys like, even in the room right now, you just log in and you are watching TV or something?
No, I'm sure we're all here.
Okay. All right, then let's talk about the control of breathing. Now this actually does relate to lab 11. I have not seen the video because what I'm trying to do is I'm trying to get to more normal working hours. I've been going crazy overtime for 4 weeks and I'm kind of getting fatigued. So I'm trying to get off early, or not early, I wish, but get off on time and kind of get back to a normal schedule. So I have not seen it, but I'm just going to tell you what is really important in lab 11. When we would do this used to be an individual lab report lab. I would get 6 volunteers, 3 who are regular exercises, 3 who are not. They would take their resting pulse rate. Then I'd have them run 2 laps around the track, come back in and start timing and everybody's got to be doing this as part of the homework, by the way, not running around the track, but just do some sort of nonstop exercise. And that's really key. You can't rest, all right? So like if you just have some steps at your house, just climb up, climb down, climb up, climb down. Just keep doing that, and do that for probably at least 5 minutes, maybe even longer. But then you just time to see how long it takes you to come back to resting heart rate. And that's where we have to talk about chemoreceptors and various portions of your brainstem, vital reflex centers that are very important for controlling heart rate. So we have mentioned this when we were talking about the brain, that in the vital reflex centers, we had the respiratory rhythmicity center as one of the 3 vital reflex centers. And we can even split that up into the posterior respiratory group. The old name dorsal was still stuck, dorsal respiratory group, and the ventral respiratory group, and how it's working together with the respiratory control centers in the pons that include the apneustic and the pneumotaxic center. Remember when we talked about this with the brain, it's like the apneustic center is constantly got the foot on the gas, and the pneumotaxic center is continually pumping the brakes so you don't hyperventilate. I think your textbook calls the pneumotaxic center the pontine respiratory group. All right, so that figure is on that slide. And then we have chemoreceptors. So this is why when you come back from running the track a couple times or doing stair climbs for a good 5-10 minutes. Why it is that both you're breathing really hard and your heartbeat is going fast. And that is because you have sets of chemoreceptors that are sensing the increased acidity when you are working out. And that's what is causing you to hyperventilate. All right, so we have two main categories of chemoreceptors. We have peripheral chemoreceptors, that means it's outside your brain. And those are very close to the baroreceptors, because we have baroreceptors in the aortic arch and the carotid sinus, and right next to them, right next to the baroreceptors in the carotid sinus are the carotid bodies. And right next to the baroreceptors in the aortic arch are the aortic bodies. And they use the same cranial nerves. Those are the Roman numerals you see on the first bullet there. And then you have the central chemoreceptors that are really close to the fourth ventricle in the medulla oblongata. Now, all of them are giving your brain information about, and that's the little P's there, PCO2, pH, PO2. Okay? But if we look at all these chemoreceptors together, we have to come to the conclusion that your bodies are way more sensitive to levels of CO2 than it is to oxygen. Fourth bullet, why? Because of this. CO2 influences pH, oxygen does not. Okay? And so, out of all those chemoreceptors, the only one that actually senses oxygen is the carotid bodies. And when partial pressure of oxygen is within normal ranges, they don't even sense oxygen. The only time the carotid body sends an alert to your brainstem is if the partial pressure of oxygen drops too low, like below 60 millimeters of mercury. Okay? So really, and usually that your partial pressure of oxygen is not going to be that low. So basically, on a normal, day-to-day basis, your brain doesn't even give a rip about oxygen. It's all about CO2. But here's the thing: is if you're really acidic because you ran a couple of laps around the track, you've gotten so acidic, you're doing this, trying to blow off CO2 because you're so acidic. Both of these are really high. So if you blow off the CO2, your acidity goes down. But what happens at the same time? You're getting more oxygen. So even though oxygen debt is a real thing, it is really almost, you correct oxygen debt almost as an, oh, by the way, secondary effect, because you're too busy getting rid of the acidity. Does that make sense? Hopefully. All right, so this is showing you the chemoreceptors on slide 55. We're doing really good. And then on slide 56 is showing you the central chemoreceptors in the medulla oblongata. And here's the key. The reason why it's close to the fourth ventricle is the central chemoreceptors are only sensing the pH of the cerebral spinal fluid. The central chemoreceptors are not even caring about PO2 or PCO2. It's all about pH. And that's why your body is actually more sensitive to levels of CO2 than oxygen. Okay? So slide 57 is from your textbook showing you the greater effect of CO2. It's affecting everything. Only on the far right there, far left, upper left where it says arterial blood, it says only if the partial pressure of oxygen drops below 60. If it's not for that, oxygen has nothing to do with that figure. That's all about CO2. All right, and then let's talk about the pulmonary stretch receptors. And this has to do with, so on page 68 of your lecture packet, you can see how the ponds. And up at the very top of page 68 is the RCC. Then just below that is the RRC, part of the vital reflex centers. But what this slide here is doing, talking about the Herring-Brewer reflexes at the bottom, I'm going to have you make sure you write something at the bottom of page 68. Because it makes it look like these stretch reflexes are happening all the time with you. And that's not true. Okay, and that's what the correction that I wrote at the top of this slide 58. And that's, yeah, those reflexes are working in newborns. But as you start growing up, and by the time you're adults, you have very little Herring brewing reflexes. Those really only kick in at, and this is what I would write at the bottom of page 68, in adults, only at the edges of vital capacity. Only at the extreme edges of vital capacities do these reflexes work in adults. What's vital capacity? That's what you're going to learn after next week's lab on respiratory physiology. Vital capacity is the maximum amount of air that a living person can move in and out of the lungs. So take the biggest deep breath you can. That is where these reflexes will start kicking in. Squeeze all of that out. And just about when you're done, that kicks in. That's the only time these reflexes are kicking in adults, is at the edges of vital capacity. Okay? So in the normal tidal breathing, that's just breathing normally, these are irrelevant. They are only working in newborns, okay? So they do help in preventing overinflation and overdeflation of the lungs because they will kick in at the edges of vital capacity. Okay, that's respiratory physiology, and now turn to page 69 of your lecture packet, and we are going to cover acid-base metabolism. Again, this does include the respiratory system, but it includes more than that. First, some definitions. Remember that an electrolyte it could be an atom or even a small molecule where the total number of protons do not equal the total number of electrons, so that atom or small molecule has a charge, like bicarbonate ion. You know, there's 5 atoms there, but you add up all the electrons and all the protons, and it has 1 less proton. So that is an ion. In the case of an acid, it is an electrolyte that releases hydrogen ion. That's why this right here in the middle is carbonic acid. Okay? Let me move this. Carbonic acid is an acid because when you put it in water, poof, it does this and releases hydrogen ion. A base is an electrolyte that tends to suck them up. So in this case, bicarbonate can actually be a base because when you put it in water, it's going to suck these up and turns it into that. If you get enough of a base in a solution, you will have very little free hydrogen ions. And remember, you need to have some free hydrogen ions in any solution. Water has it. And if you look at the pH scale that we talked about in the very first lab, back in the beginning of the semester, pure water does have a concentration of free hydrogen ion. But look at that pH scale on the far right. Look, go to the very top of that pH scale at the very top and notice it is not free hydrogen ion, it's hydronium ion. That free proton glommed onto a water. Okay. Now the body has a very narrow range of pH, and you can see that on the top of page 69. And it centers around 7.4. Okay. You go neutral, you go to 7.0 and you feel like crap. Okay. We work in a very narrow range of pH. At neutral, you're feeling sick, and you will be hyperventilating something fierce if you're neutral, because your body wants to be slightly alkaline, 7.4. All right, so let's talk about, before we talk about how our bodies deal with acid-base imbalances, we got to talk about the sources of hydrogen ion in our body. And we actually covered it in lecture exam 1. If you're doing aerobic cellular respiration, you're going to have this, that's going to contribute to acid. If you do anaerobic respiration, you're going to generate lactic acid. And lactic acid also releases hydrogen ion. So whether it's aerobic or anaerobic, you're going to increase acidity. Some other things that you may not have known, that this was actually covered in Lab 8, even though we didn't have a quiz for Lab 8. When there is poorly controlled diabetes mellitus going on, there is above normal lipolysis happening. And if you have above normal lipolysis happening, you have an above normal beta oxidation. And remember, beta oxidation is not one reaction, it's several reactions. And one of the intermediate steps in beta oxidation are ketones, which is why diabetics, when they're doing too much of this, have diabetic ketoacidosis. And that is giving you a clue. That name, diabetic ketoacidosis, is that ketones are acidic. And so if you're doing too much beta oxidation, you're going to get acidic. And that's mentioned there on point number 3. Also, when you're metabolizing amino acids that have sulfur in it, one of the byproducts, and it's an intermediate step, it doesn't stick around for very long, is sulfuric acid. And sulfuric acid is a really strong acid. And then when we talk about the metabolism of nucleic acids and nucleoproteins, because they go hand in hand, those phosphate groups will eventually come off as phosphoric acid, which is also extremely acidic. So when you have these things, it's just a normal response of living. You're going to metabolize amino acids with sulfur. You're going to have to recycle and make various nucleoproteins. You're going to have points where you're going to liberate hydrogen ions. So our response to acid base imbalance we're going to organize it in either immediate or delayed responses. And the immediate one that you have to know for lecture exam 4 is buffers. Chemical buffers. And the definition is right there. You can add either acid or base to a solution. And if you have a buffer in there, it is going to level out the pH change as you're adding the acid and base. So let's look at this. Slide 62. Okay, so if you have a strong acid going into solution, you can have a weak base that can act as a buffer that will form a weak acid and a salt. And that's in the box there. If you add a strong base, you can add a weak acid to form a weak base in water. Let me explain this graph that's on the bottom of slide 62. Now look at that graph on the bottom of slide 62. And it's talking about adding base. And the reason why they're talking about adding base is because the pH is going up as you add base. Now, if you don't have a buffer, this graph is going up linearly. You just add base, pH is going up in a straight line. Now look at that graph on the bottom of the slide. It looks like this. And this is with buffer. And I like that S-shaped line. What that S-shaped line is showing you, I should have it go down. What that S shape is showing you is that the buffers have a functional range. And that's the red line. Before the red line, the blue line is the same as the black line. After the red line, the blue line is the same as the black line. But in that range, in that functional range, the line is flat. That's why it looks like an S shape. Okay, that slide that has that graph on it is showing you the bicarbonate buffer system. And it can go either way. You could buffer both an acid and a base. And then the phosphate buffer system, this is on slide 63, parallels it. Okay. Now notice that these immediate buffers are working in both inside the cell, ICF, and outside the cell, ECF. The phosphate buffer system is pretty crucial in urinary physiology, when I have some pictures of that. Also, you should know that proteins, because they have carboxyl groups and amino groups, are also really good buffers. So plasma proteins, that's one of the things that can cause your plasma pH to be relatively stable, because you got all these plasma proteins in there. And these amino groups, and you can see this in the figure on the bottom of slide 63, can either soak up excess hydrogen ion or give some off in case things get a little basic or alkaline. So proteins can go both ways. All right, then let's talk about delayed responses. And the two that you need to worry about is what we just covered. You just alter How much CO2 you blow off, you can either increase it or decrease it depending on your pH. If you're too acidic, you're going to hyperventilate. If you're too basic, you're going to slow down your breathing and build up CO2 to offset the basic pH. That's pretty quick, but not as immediate as buffers. And this is showing you the same thing here from your textbook on slide 65. And then the other delay mechanism is the urinary system. And if you look at the slide on slide 66, you see that there is an exchange between the filtrate in the kidney tubules of the nephron and the peritubular capillaries. And again, you don't, in a similar way of the chloride shift and the reverse chloride shift, you do not want to have an accumulation of positive ions on one side. So if your kidney tubules are secreting out a positive ion, and it's usually potassium, it's going to reabsorb sodium. Okay. Now, if you are acidic, what it'll do is it will not secrete out as much potassium, and in exchange for potassium, it'll actually secrete out hydrogen ion. That way it's all balanced between a positive ion and a positive ion, but now you can get rid of acidity. And when we do Lab 15, we would normally have 9 volunteers. Every semester, a majority of the volunteers will have acidic pH because they get rid of acid. Okay. Now that free hydrogen ion is not going to stay free hydrogen ion. It's actually going to lom onto ammonia, which is in urine or at that point filtrate, and it becomes ammonium ion. Okay. And that just can't get back into your tissues, and that's why it stays in the urine. But it was the acid, now it's hydronium ion, and ammonium ion, and that's going to just go out in the urine. And just also remember that the phosphate buffering system is also helping to keep the pH from changing too much. And then you just finish up with figures from your textbook showing you again this ion exchange. On slide 67, this is on the left side of the tubule cell, and the sodium is exchanging for a hydrogen ion, which is acid. Okay. And then on this slide 68, you can see where the phosphate buffering system will help keep the acidity of the filtrate from getting too caustic. And then this is also showing you how on slide 69, how that hydrogen ion will lom onto ammonia, forming ammonium ion. Okay. Last slide, slide 70, is showing you the drop deadline. Remember, the drop deadline is 4/27 at 7 days. And it says with a W. Actually, if you haven't heard yet, because of the extraordinary circumstances of this semester, and I haven't heard anything that has changed this. I was told that all W's this semester are going to be EWs, not just straight W's. Yes, it says that on your transcripts, but an EW is called an excused withdrawal, which means it doesn't count against you in applications to programs, and it doesn't count against you as a strike as one of your attempts at physiology. So it is basically, it's there to show that you were registered in the class after the first drop deadline, but it doesn't count against you. They usually were reserved EWs for something serious, like seriously medical happens in the middle of the semester. You're after the first drop drop deadline, and you had a bad car accident and was in the hospital for a month, well, then you're going to get an EW. That's kind of things like that. So that's because of this semester, that's what all the withdrawals are going to be on the transcripts from the semester are EWs. But that is Monday. If you are registered after Monday, then you got to get a grade. Okay? Any questions? So we will have one more lecture on lecture exam 4 on Monday, and then Wednesday of next week is lecture exam 3. Okie dokie.
Do you have picture? Can we do pictures again?
Oh, yeah. Everybody see that? That's the left bank. That's the middle. The graph on the buffers.
Thank you.
Okay. All righty. I will post this as soon as it's done. See y'all later.